Hi, welcome back. I'm Kate Watkins, and it's my pleasure to be chairing session 6 on the first day of this conference. And our speaker in this session will be Soo-Eun Chang, who is at the University of Michigan and Michigan State University. I'm sure she needs no introduction, but I'll give a brief one. So Soo-Eun is the director of the Speech Neurophysiology Lab. She's an associate professor in the Department of Psychiatry and the Rosa Casco Solano Lopez Research Professor of Child and Adolescent Psychiatry at the University of Michigan. Soo-Eun is also an adjunct professor at Michigan State University in the Department of Communicative Sciences and Disorders and the Department of Psychology. Frankly, I don't know how she manages to do any work with all of those departments. You'll be aware of her groundbreaking work where she has used MRI brain imaging to look at the differences in the brains of children who stutter, and she had some lovely work when I first became aware of her work around 2008, looking at differences between children for whom stuttering persists and children who are considered to be recovered. And I'm really pleased to know that she's been funded well by the National Institutes for Health to carry on this really important longitudinal work. She's also doing work very similar to that I talked about earlier today, looking at the effects of neuromodulatory techniques to augment therapies for people who stutter. And today I think Soo-Eun is going to talk to us about the imaging work in a very large sample of children that she's been carrying out over the last few years. The title of her talk is Grey and White Matter Developmental Trajectories Associated with Childhood Stuttering Persistence and Recovery. So over to you, Soo-Eun.
Hello, everyone. My name is Soo-Eun Chang, and I am an associate professor at the University of Michigan. Today, I will be presenting recent work from my lab investigating brain structure differences in children who stutter. Before we dive in, I thought I would put things into context on what we are trying to do when conducting neuroimaging research in stuttering. As we all know, stuttering is a complex disorder. And like most complex disorders, it is diagnosed based on its behavioral symptoms rather than with some kind of objective marker, like a blood test, for example. Also similar to other complex disorders, the cause of stuttering has been proposed to be multifactorial, with genes and the environment playing roles in how the behavior of stuttering is manifested in an individual. Genes and environment interact to affect things like neural function, neural circuit formation, and connectivity among critical regions, which can all be captured through various neuroimaging techniques. Genetics is obviously very important and can provide us information on basic biological processes that might be affected. But these basic biological processes at the level of proteins can seem very far away from the complex behavior we are interested in explaining. Brain anatomy and function, on the other hand, provide a more proximal window into how the behavioral manifestations of stuttering may arise. Critical regions and connections in the brain have been studied and mapped out in the context of typical neural development. And by examining where and how things differ in people who stutter, we can deduce what functions and connections are relevant to stuttering etiology. This can lead not only to a better understanding of the nature of stuttering, but can pave the way for neuroscience-based intervention of stuttering in the future. In this presentation, I will first explain what I mean by gray and white matter, and briefly review aspects of brain development related to gray and white matter growth, focused on regions relevant to speech and language. I will follow that with a discussion of how examining brain structural measures is relevant to studying neural bases of stuttering. then lead into the main study that was part of an ongoing longitudinal study of childhood stuttering. I will outline the procedures used in that study and explain the structural MRI data analyses that were applied, in this case, voxel-based morphometry. I will then discuss the implications of the findings, including a little bit about possible neural mechanisms of recovery during childhood. When talking about gray matter, we are talking mostly about the outer cortex and deeper structures such as the thalamus and basal ganglia that you can kind of see in this section, coronal section shown here on the top right corner. These areas contain cell bodies and dendrites of neurons that are shown here. The cell bodies can be thought of as the main information processing area, whereas the dendrites form synapses with other neurons, which form the basis for neural communication and circuit formation across the brain. In humans, the full number of neurons are reached by the 25th week of gestation, and dendrites begin to develop a few months before birth and are primitive at birth. During the first year of life, dendritic processes develop on each cortical neuron to establish what is an amazing number of connections that each nerve cell makes with other neurons, and this can range between 1,000 and 10,000. So you can see here that at birth, these connections are sparse, but you see much more here at 6 years of age. The difference between the 6-year-old and the 14-year-old is that in the 14-year-old, now there seems to have been some pruning that occurred. And indeed, this is the case. Some connections are strengthened while others are pruned away. And this makes the neural function much more efficient. Communication velocity among different areas is supported through what's called myelin here. I don't know if you can see my cursor, but the myelin insulates the axons and increases the speed of transmission of nerve impulses. And this myelin has a whitish appearance, and so the areas with collection of axon bundles that are covered with myelin are called white matter. In most areas of the brain, gray matter volume shows an almost linear decrease across the lifespan. So you're born with more than you need, and many are pruned away, or in some cases, gray matter areas are taken up by white matter growth. White matter, on the other hand, shows continued increases in volume up to one's 30s and 40s, followed by decreases. I just mentioned that gray matter has an almost linear decrease from birth to old age. However, there are some exceptions to this, most notably in areas relevant to speech and language. By now, it's an old study here, but you can see that gray matter measures continue to show growth well into early adolescence in the case of the left inferior frontal gyrus and the auditory regions bilaterally. And in the case of the left posterior STG, which includes the association auditory region that's very critical for speech and language processing, the gray matter density did not start to fall until much later in life compared to other regions. This indicates that cortical speech and language relevant areas have a more protracted period of growth compared to regions supporting other basic functions such as vision or limb motor function. What about white matter? Another classic study, but this one showed that among major white matter tracts, the left arcuate fasciculus was one of the exceptional tracts that seemed to be still increasing up to later adolescence. So why study gray and white matter in stuttering? Well, the neuroanatomical differences reflected in gray and white matter volume changes may be associated with functional consequences relevant to speech. White matter changes may suggest deficits in structural connectivity that in turn affect efficient neural communication among critical areas. Also, examining the changes in developmental trajectories in gray and white matter may provide clues to critical regions and what the developmental periods are that are critical and relevant to stuttering, persistence, and recovery. In the next few slides, I will talk about an ongoing longitudinal research program that enabled the current study of gray and white matter growth in children who stutter. The longitudinal neural imaging study of stuttering at Michigan has been going on for about 10 years. Children are recruited from the community, and from each child, we collect a comprehensive list of behavioral assessments, including standardized assessments measuring their speech and language development. We test their IQ. We also collect a detailed developmental history, their socioeconomic status. We video record speech samples to allow offline analyses of language development and dysfluency analyses. We then, of course, collect structural and functional MRI data. All of the MRI data collection and most of the behavioral testing are repeated up to 4 times, 3 times for most children, with an inter-visit interval about 1 year. We've had 2 cohorts complete all parts of the longitudinal study procedures to date. And this shows a summary of the timeline that occurred for the second cohort. Each of these gray blocks indicate when all of these assessments and MRI scanning occurred. All children who stutter that entered the study met the criteria for stuttering based on their recorded speech samples comprising conversation and narrative speech, from which we calculated their SSI scores and percent SLD measures. And we also considered parent report and clinician impressions. And because we saw each child every year, we could repeat the speech assessments and could determine whether they recovered or persisted during the period of up to 4 years post-initial visit. If a child showed 2 consecutive visits showing significant decreases in SSI, and that scored below very mild, and the parent could confirm this by reporting absence of stuttering at home, and considering clinician report, these children were determined recovered. The persistent children, on the other hand, were those that continued to have very mild or higher severity levels at later visits. And even if they showed a relatively low SSI and SLD values in the lab at one of the visits, if the parent indicated continued stuttering at home and other settings, we considered them to be persistent. Because this is an MRI study, we set up a detailed training protocol for mock MRI sessions to desensitize the children to the MRI environment. And because our youngest kids could be as young as 3 years of age, these training sessions were critical in ensuring that each child was trained and ready to participate in the MRI session with minimal anxiety and discomfort. These training sessions would involve not only having them be exposed to the sights and sounds of the MRI scanner, but also practicing to be still for periods of time and making them aware of what happens if they move. So here you can see what we would show the child. Every child basically after getting participating in an MRI session would be able to take home a picture of their brain, and we would show them what happens if there is movement. Would you rather get a blurry picture or a clear picture like this? They also had a chance to try on helmets that fit like MRI head coils. They also practiced sliding in and out of the mock scanner bench, etc. So these were a series of activities that were designed to be fun and at the same time serve the purpose of training and readying them for the real scanning session. The staff also had a chance to assess, and they were able to have a pretty accurate idea of the readiness of each child through these sessions, so that we would not schedule an MRI session for any child that seemed overly anxious or otherwise not ready to participate in the actual MRI experiment. These mock training sessions were repeated as needed. If a child did pass the mock training with success, they then participated in the radiology visit, where they were greeted with colorful screens that effectively hid any scary-looking parts of the scanner, and they thought they were entering some sort of ocean adventure. The clinician sat at the side of the child at all times to ensure comfort and to remind, in some cases, for children to keep still. So what were some of the study questions posed for the brain structure study that I will be talking about today? We asked what brain anatomical differences in terms of gray and white matter volume were present in children who stutter, and whether these differences are consistent across development. We also asked whether any of these volumetric changes correlate with stuttering severity. We also asked whether gray and white matter development differentiate persistent stuttering from recovery. Importantly, we thought the current study could overcome some of the limitations of previous studies. By examining both gray and white matter in the same study, we expected to have a more comprehensive view of structural differences in children who stutter relative to age-matched peers. We also were able to examine both cortical and subcortical regions, including the cerebellum, which has not been the case for many previous studies. Another strength of the study was that we were able to conduct a longitudinal analysis that enabled examination of age-related changes and developmental trajectories, which was not possible with cross-sectional studies in the past. This is also the largest study to date that included close to 100 children in the control and stuttering groups. Each of the group had about 100 kids, and this is the largest such study to date in our field. Based on previous empirical findings and theoretical perspectives, we hypothesized that children who stutter would differ from age-matched controls in areas that are part of largely 3 neural networks. This included the frontal temporal network on the left hemisphere, which supports auditory motor integration. Second is the basal ganglia thalamocortical networks that support motor initiation and sequencing, as well as rhythmic or interval-based timing. Cerebellar cortical circuits were also of interest, given that the cerebellum is a major structure involved in the generation of the forward model that plays a role in prediction of sensory feedback and detection of error and fine movement adjustments. Together with basal ganglia, the cerebellum is also a major structure involved in temporal processing. Previous studies had found evidence supporting deficits in all of the labeled areas shown here and connections among them. We expected to replicate these findings in our child cohort, especially for the persistent children. We further hypothesized, based on our previous diffusion tensor imaging study, that the recovered group would show age-related normalization in the left arcuate fasciculus, a branch of the superior longitudinal fasciculus that interconnects frontal areas to the temporal parietal regions. In addition, based on a functional MRI study that showed deficits in the basal ganglia thalamocortical network related to rhythmic timing tasks in children who stutter, which indicated deficits in intrinsic timing control, we further hypothesized that recovered children may have a compensatory growth involving the cerebellum to compensate for deficits in the basal ganglia network. For this study, we used voxel-based morphometry, which allows automated extraction of regional gray and white matter volume and objective group-based analyses on a whole brain basis. Basically, first we segmented the gray and white matter in the brain into probability maps. They were used to normalize individual brains into a standardized space. The transformation matrix derived from this was applied back to the probability maps, and this generated modulated gray matter and white matter, which reflect the volume of each fossil in the native space. In terms of participants, everyone was recruited from the local community in the East Lansing, Michigan, and surrounding counties. These were not based on clinic samples. Of the total 506 screened so far, 276 have completed the behavioral testing to date. We were greatly affected by COVID and have not been able to collect data since last March. Among the 276 that completed this speech language testing, 199 participated in the MRI session. Of these, we were able to use structural MRI scans from 189 children, that's 94 children who stutter and 95 controls, who contributed a total of 462 longitudinal scans. You can see the breakdown in more detail here. We further divided the stuttering group into persistent and recovered groups based on speech data analyzed from their later visits. 72 were determined to be persistent and 22 recovered. Just a really brief mention about the longitudinal data analysis applied here. We used the sandwich estimator method that has been validated and proven to be accurate and efficient, and has been used in other longitudinal neuroimaging studies. The model estimated the main effective group. In this case, we were interested in the persistent stuttering group, recovered children who stutter, and controls. We also looked at interactions, which included group by sex, group by age, group by quadratic age and interactions. We included socioeconomic status, IQ, and total cranial volume as effects of no interest to control for their effects. We also examined the effect of SSI within the stuttering group to examine whether stuttering severity correlated with changes in regional gray and white matter volume. Okay, so now to some results. First, let me present some overall group contrast results based on the comparison between persistent children who stutter and controls. Now, in the middle column here, note that I'm showing here the results based on the whole age range of the participants included, which was 3 to 12 years of age. The color blobs you see, which are consistently blue here, indicate areas where controls were significantly greater in gray matter volume compared to persistent children who stutter. These included the bilateral thalamus and the left ventral premotor cortex in the overall contrast based on all of the subjects. When examining the results within smaller age ranges in younger and older subsamples, so the younger being 3 to 5, older 6 to 12, we can see that in the younger group, the difference was significant in the putamen, bilateral putamen, while the thalamus became more different in the older group. The left ventral premotor cortex, while present in each age group, was subthreshold in the subgroups, only reaching significance with the bigger sample combining both age ranges. Some other areas that showed up in the persistent group versus control contrast included the right caudate and putamen and the right cerebellar lobule 6, the latter being more evident in the older group. Again, these were all areas where the persistent group showed significantly reduced gray matter volume compared to children who do not stutter. What about correlations with stuttering severity? Interestingly, 2 of the regions that were found to be significantly reduced in gray matter volume in the persistent group compared to controls that we just saw, the putamen and the right cerebellum, were also areas that showed a negative correlation with SSI. In other words, the lesser the gray matter volume in these areas, the higher the stuttering severity was in the persistent group. Moving on to the contrast between recovered and controls, this did not show any interesting significant findings. Some were present, but most were subthreshold. The contrast between the recovered and persistent groups showed differences primarily in the cerebellum. The right cerebellar lobules 6 and 7 in particular showed age-related differences, as shown in the scatter plots on the right, both indicating that the recovered group had greater increases in these areas with age compared to the persistent group. Okay, so I've gone through already a lot just talking about the gray matter. We haven't even talked about white matter yet. So just to sum up what we have so far from the gray matter volume results and putting them in the context of the 3 networks that I mentioned at the outset of the study, we found significant decreases in gray matter volume in persistent children who stutter relative to controls, the fluent children, in the ventral premotor cortex, basal ganglia, specifically putamen, and thalamus regions, as well as cerebellum. Furthermore, 2 of these areas, the basal ganglia, the putamen, and the cerebellum, showed negative correlation with stuttering severity. In other words, these were areas that not only had reduced gray matter volume in the persistent kids, but were also areas varying with stuttering severity. The right cerebellar lobule 6 was particularly interesting because this was an area that seemed to show increases in the recovered group compared to the persistent group. What about white matter? Well, examining white matter volume might give us glimpses into the structural integrity of important white matter tracts, such as the left SLF shown here on the top, interconnecting the ventral premotor cortex IFG region to the posterior STG region. This is an area, this is a white matter tract that was often reported to be decreased in white matter integrity based on previously reported DTI findings in the literature. So what did we find? Well, this slide shows results from the contrast between the persistent and the recovered children who stutter, both the main group contrast in the top and the age by group effects. They're all blue, meaning that the recovered children were consistently greater than persistent children in these areas in terms of white matter volume. This was true in the overall group contrast, as I mentioned already, as well as when we looked at the age effects. In other words, the recovered group showed a much steeper slope of white matter changes, white matter increase with age in the left superior longitudinal fasciculus area. This age effect slice shown here, this is a fairly large cluster, and this likely includes not only the SLF, the superior longitudinal fasciculus, but also some descending tracts that carry fibers from the cortex down to the subcortical areas and the brainstem. You can see here that the recovered kids seem to exceed even the control group, which is shown in the blue line here. Green are the recovered kids, red are the persistent kids. So if you see the slopes of this, it seems the recovered kids exceed not just the persistent kids, but even the control group. So indeed, if we look at the contrast between the recovered group and the control group, we saw that the SLF showed up to be significant, but also other areas showed up, including the bilateral cerebellar dentate region, as shown in the bottom here. This area contains efferent fibers that ascend from the cerebellum to cortical areas via the thalamus, which could support fine motor adjustments in response to speech errors, a function well known to be supported by the cerebellum in response to mismatches occurring between expected and actual sensory feedback generated by one's own movement. In terms of correlations with stuttering severity, the left superior longitudinal fasciculus and descending tract, that area showed significant significant negative correlation with SSI in the persistent children who stutter. Together with the fact that this was also an area that showed significant age-related growth in recovered kids, as shown here on the right, that exceeded that of controls, white matter growth in this area may play a role in helping to produce more fluent speech and achieving recovery. So a summary of the white matter findings. We found that the left SLF was significantly greater in white matter volume in recovered children relative to persistent children, and furthermore, this difference became greater with age. The recovered children who stutter showed a steep growth curve in the left SLF, as well as the left descending tracks. There was also a negative correlation between SSI and white matter volume in this area. Interestingly, the recovered group also showed increases in the bilateral dentate nuclei compared to controls, which may indicate a compensatory growth involving greater ascending white matter from cerebellum to cortical areas. These will need to be confirmed in future studies, perhaps in combination with DTI and functional MRI studies. Putting these results in context of a recent theoretical perspective outlined in this review paper by Chang and Gunther, here are three distinct but not mutually exclusive loci of impaired neural processing was proposed to occur to lead to stuttering. First is the impairment in the basal ganglia proper, shown here, indicated with PDS1. Second is the impairment of axonal projections from the cortex, basal ganglia, and thalamus, shown here with red lines and indicated as PDS2, and impairment in cortical processing, shown as PDS3 here. In the current VBM data set that I just presented, we seem to show potential support for neural structural changes linked to all three of these loci. In PDS1, or basal ganglia proper, we saw that the putamen was significantly decreased in gray matter volume in persistent children who stutter compared to controls. We also saw that white matter volume in areas containing descending tracts from cortical motor to basal ganglia and brainstem areas were decreased in persistent children who stutter, supporting PDS2. Possible impairment in cortical processing, as implicated in PDS3, was also found in that the ventral premotor cortex gray matter volume was significantly decreased in the persistent group, and connections between this area and sensory regions via the SLF was decreased. However, there is one more thing that was found in the current data set that was not included in this theoretical framework. Now, this is a figure based on another simpler figure published by Kotz and colleagues, where they posit that cortical basal ganglia, thalamocortical circuits engage in sensory motor temporal processing. Here, the thalamus is placed at the center for integrating cerebellar inputs that carry information on the temporal structure of the sensory object or sensory feedback that is linked to self-initiated actions. And this signal, this information for the cerebellum goes via the thalamus, conveyed to the SMA, supplementary motor area, as a pacing signal. Kotz and colleagues posit that this subcortical connectivity between the cerebellum to the basal ganglia and SMA regions via the thalamus is something that may enable automatic adjustments in sensory motor timing errors. So what do all of this have to do with the data that were presented today? Well, many of the important players in this model involved in temporal processing of sequential movement were found to be significantly reduced in gray matter volume in persistent kids who stutter. And it is intriguing to speculate how deficits in temporal processing could be at the core of persistent stuttering, which also affects stuttering severity. The right cerebellum and putamen were shown to be negatively correlated with SSI. In recovered cases, we not only saw increases in cerebellar dentate regions, but also greater white matter volume in tracts that interconnect auditory motor cortical regions as well as cortical subcortical areas. This may indicate that normalized auditory motor connectivity as well as better temporal processing may play a role in recovery from stuttering. To wrap up, today I presented data from the first longitudinal study of whole brain morphometry in childhood stuttering. We found regional gray matter and white matter volume differentiated among the persistent children who stutter, the recovered kids who used to stutter, and children who do not stutter. Persistent children who stutter had decreased gray matter volume in the basal ganglia, the thalamus, right cerebellum, left inferior frontal gyrus / ventral premotor cortex, suggesting that primary deficits in motor timing and sequencing may be involved in persistent developmental stuttering. And this may also indicate relevance to stuttering severity. Recovered children who stutter exhibit heightened white matter volume increasing with age, which exceeded even controls. And this pattern was shown in the left superior longitudinal fasciculus, some descending white matter tracts, and the cerebellar dentate ascending tracts. These results suggest a possible compensatory role of the cerebellum in movement control and a normalized connectivity of the dorsal tracts that may improve auditory motor integration. Given that volume changes affecting major structures given that volume changes were found in areas that were involved in sensory motor temporal processing, as was shown in the previous slide, normalization in some of these areas in recovered children might suggest that engagement of interventions that involve timing and rhythm might be beneficial. However, much more research is needed in this area. I'd like to thank my lab as well as all the parents and children who have participated in this research. And I would like to also acknowledge the funding sources, NIH, NIDCD, and private gifts that I have received that have allowed us to do all this work. I also want to announce that we have at least a couple of fully funded postdoc positions that are available. So those who might be interested in conducting new fMRI. We also have neural oscillation studies that have already been funded and are being started right now. And we have clinical trials going on with brain stimulation studies. Those who might be interested in any of these endeavors, I encourage you guys to email me. So thank you for your attention, and I would be happy to answer any questions. Thank you.
Okay, I think we've lost Kate. Gemma, are you there? Gemma, Kate, are either of you there? Okay, so Soo-Eun, it looks like we have, I think we might have Gemma now.
Yep, hold on 1 second.
Thank you, Gemma. If you could take over with Kate just being missing at the moment.
Lovely. Okay, so let's have a look at. Oh, I think I've disappeared as well. Soo-Eun, thank you for such an interesting talk. Really fascinating. Oh, and there's Kate. Thank you.
I'm really sorry. I don't know what happened, but this happened. I had to refresh my screen. But anyway, I won't take up time. Shall I go ahead? Thank you, Soo-Eun. I mean, that was just fascinating talk, and blown away by the amount of data you have. And I fully appreciate just how difficult it is to collect all of that data in anyone, but in children, an extra challenge. I think you've probably had a chance, hopefully yourself, to see some of the questions in the Q&A. But I'll start with the top one, which says, excellent. Oh, new ones. Excellent study. What were some of the reasons that the children did not remain in the study? Was it related to recovery?
So we would not, so there were very few cases where people would stop coming to the visits because they had recovered. So we don't offer therapy. This is not a clinic. And they fully understand at the outset of the consenting process that we're specifically interested in monitoring them for a number of years, and that the child might continue to stutter or recover from stuttering, and we're interested in both of those trajectories. So it was very rare for families to stop coming just because the child recovered. Now, some cases of attrition do occur, obviously, in a longitudinal study like this, and these primarily were due to families moving away, or in some cases, kids getting braces, for example, and so we couldn't include them anymore for MRIs. So a lot of various reasons, but we're happy to say that we've been quite successful in retaining a lot of these participants, and our attrition rate has been quite low. So that's been great for us. And in fact, a lot of these families, even when they age out of the studies, we have about 20 or more families that came back to us. Now the kids are in adolescence, and they're asking, can we come back for a different study? Is there something we can participate in? So that kind of speaks to my staff and the speech language pathologists, who do a wonderful job in really engaging the kids. And they truly think that they're little scientists who are participating in something helpful and interesting. And we definitely make it so that it's fun for the kids to participate in. So yeah, that is all to say we do our best to keep our attrition rates low.
Okay, I think some people are saying they can't hear the answers. I don't know what to do about that, but maybe some of the tech support could give us sort that out in the background. Next question I'm going to go to is sort of related a little bit, I suppose, because we all appreciate how much harder perhaps it is to recruit the children in that recovered group for reasons you've already outlined. So the question is whether there were any differences, perhaps predictors in the recovered group, the children that went on to be classified as recovered, that you could use perhaps as predictor of whether they would recover or be persistent, some sort of like whether they were more mild or more severe on the SSI at earlier stages.
So I think the general understanding is that the severity at onset does not really predict persistence or recovery. However, we are finding in our group of recovered kids that they do tend to be a little bit more on the milder side, and we definitely don't see as much prolongations or blocks, for example, in this group. But I have to caution and say that our recovered group of kids, which was only about 20 kids, it's not a large group. So we have to kind of look at this further. And the reason why we have fewer recovered than the persistent is that although we try to recruit the kids as young as possible and close to stuttering onset as possible, we have to kind of balance that out with who can actually do the MRI. So we end up getting kids with first visit, they're between 3 to 6 years of age, but the ones that end up giving us successful scans tend to skew a little older. So if you're older in the beginning, you're more likely to be persistent. So that's probably why we have more persistent kids in our cohort than the recovered kids. But yes, we do need to collect more information from the recovered kids. So far, based on the small sample we do have, it does seem that the recovered kids trend having milder stuttering than the persistent kids.
Okay. So that's an interesting question. I mean, it's great to have that kind of data. Relatedly, popular question here. Hi, Soo-Eun. Nice talk on a complex topic. My question is about the cerebellum, why its function is necessarily compensatory.
Right. I think that's a great question. So yeah, it doesn't have to be compensatory, but this is a hypothesis that we can test. So this is based on maybe the Parkinson's literature. I think a lot has been shown that in Parkinson's disease, which is obviously a basal ganglia-based disorder, there were some studies that have shown that the cerebellum seem to serve more of a compensatory role, and you see more activity in the cerebellum in some Parkinson's cases. So this is kind of guided by that kind of framework. However, I think this is one hypothesis. There could be alternative hypotheses. It could be that the cerebellum is maladaptive, but given the data that we have, it does seem like in certain parts of the cerebellum, specifically on the right side, which, as we know, it has connections to the left hemisphere, we see that in the persistent kids, reduction in that right cerebellar lobule 6 seem to be related to persistent stuttering. So yeah, and then on the other hand, in the white matter, we see increases there for the recovered cases. So it could be a combination of the fact that there's aberrant growth in the cerebellum, which could in effect be maladaptive rather than compensatory. And I can go on more about this. There's more recent findings about the fact that it's not like a dual separate loop between cerebellar cortical, basal ganglia cortical. It's not like that. There are interconnections between the basal ganglia and cerebellum. So any deficit in the basal ganglia could propagate to the cerebellum and vice versa. So it's not as simple as a one or the other. So I think good news is there's plenty of things that we can look at. But just going back to the question again, no, it doesn't have to be compensatory, but this is a framework that we're using as a hypothesis. And if the data proves us wrong, great. We have alternative hypotheses. I don't know how satisfactory that answer is, but yeah, Kate, do you have any thoughts on this yourself?
Oh, you've gone.
You're back.
Okay. Yeah, I do have some thought. I mean, we very early on had very small amount of data in some children who recovered and some persistent, and we looked at the cerebellum. And actually, I remember we didn't publish these data because they were very small numbers, but that the group that recovered actually seemed to be more different from the control group than the persistent group were. And so I kind of had this idea that maybe that difference was in the cerebellum was sort of driving some reorganization of function that led to recovery. So in a sort of neural model, the idea, I suppose, is that you kind of need to reach maybe some sort of threshold where the brain can detect that it needs to do something differently. And perhaps that was why there was recovery. But that wouldn't be consistent with your observation that the stuttering severity index was in the more mild range for that group, I suppose. My hypothesis was that they were in the more severe range, and so that had kind of forced
Yeah,
recovery.
I think depending on what specific area of the cerebellum, and looking at right and left also is
Yeah.
something we need to further look at. And and sorry to put you on the spot, Kate, but I do this because you're one of the first ones that actually did kind of dig into the cerebellar role in stuttering. So yeah, I think it's been kind of a relatively neglected area, but definitely an important structure to look at in relation to stuttering.
No, I think I think you're right, and I think this link with the basal ganglia is another one that we've kind of ignored a bit, and is clearly key, as you point out. I'm going to move on to questions. It's quite hard to see the questions as they come in because they move around. But anyway, here's one. Has there been any longitudinal research on how intense speech therapy, daily therapy for an hour, for example, can increase white matter volume on the SLF or in the basal ganglia thalamocortical loop to decrease fluency severity? Sorry, super interesting research. Thank you.
Yeah, I mean, that would be a fantastic research study to do. I don't think anything like that exists, definitely not in stuttering children. Right now, the research studies looking at intervention, I'm not sure if there have been interventions done in an intensive manner like this, except for the TDCS studies, that you've been doing, and Emily Garnett in my lab has done also. So I think we will see some of that coming out from both labs, looking at the effects of TDCS and daily speech intervention that was done intensively. So that's every day for 5 days or 7 days, and see if that changes brain function as well as white matter volume. Answer is we don't have that yet, but I hopefully we'll have that data to show.
Yeah. Another question I think you do know a little bit about is, do the white matter differences in children who stutter correlate with the differences in GPTG expression? I guess one of the genes in children who stutter compared with controls.
Yeah, so interesting. So we haven't looked at this yet with white matter. We've only looked at it with gray matter, I believe, and there was one study that was published. This is something that we need to do. And that other study that was published was only based on a subset of the data that I presented today. So we probably want to see if we can replicate those findings with this larger sample for both gray and white matter. But thank you for that question.
Yeah, I guess it's worth pointing out that we can't do it in the same individuals, that we can't look at gene expression and gray matter volume.
Well, yeah, so we do have plans to do that. So most of the kids that we have brain data on, we also have their saliva samples. So we haven't processed any of that yet. That is ongoing. So we'd like to see, who among the group of kids that we do have this mutation in these DRANA genes, and see if indeed, their expression patterns differ and they correlate with gray matter. But that's in future studies that need to be done.
Yeah, my point is I don't think we can know about gene expression in an individual in vivo, can we?
Oh, right. Absolutely. Yeah. This was based on the Alan gray matter.
To be correlational, I guess.
Right.
Yep. Great work. You and others have found reduced cortical thickness in the left ventral premotor cortex, ventral motor cortex in stuttering, but damage to these areas is not substantially implicated in neurogenic stuttering. So what are your thoughts regarding this apparent discrepancy?
Yeah, so in stuttering, just like in any other complex disorders, deficit in one area doesn't usually lead to that disorder. A deficit in one area could lead to aphasia or some other disorder, but not stuttering. And that is because in complex disorders like stuttering, it has a lot to do with the connectivity of networks of regions rather than certain, one or two or even several areas. So in neurogenic studies, since the questioner asked about that, in neurogenic studies, the highest incidence region that was found to be affected was in the thalamus and the basal ganglia regions. And these regions, importantly, have lots of connections with all parts of the cortex. So this tells you that even when there's a deficit in one area, it has to do with areas that make all these very intricate connections with other places. Now, in relation to the ventral premotor cortex that we found, yes, we find that, but that's not the only region that we find. We find differences between the stuttering and the control groups in other regions that may be interconnected with the ventral premotor cortex and the motor cortex as well. So yeah, I don't think this is a discrepancy. I'm not sure if my answer was completely clear, but Kate, maybe you can elaborate as needed. I don't know if that was sufficient.
Yeah, I think it's clear. I think the neurogenic data to me has always been a bit hard to understand.
And they're older, and yeah, so.
Yeah, I have a question here. Thank you. In your study from 2008, it was reported that children with persistent stuttering had reduced grey matter in the left Broca's area, but even lower in recovered. Has this been replicated? Did recovered children change laterality to the right? I guess that's two questions.
Yeah, so we do have to acknowledge that for this 2008 study, this was, a long time ago, and this only included about 7 in each group. We probably can't publish this kind of data set anymore. One encouraging aspect is that some subset of these findings are replicated. This aspect about the reduced gray matter in the left Broca's area and the recovered kids are not replicated. So there's that. So I don't think we have any evidence of the recovered children changing laterally to the right. If anything, we find that the recovered kids seem to have what we have written as normalized patterns of growth in the white matter in that region, as well as the interconnection with the auditory region. They seem to have reduced connectivity between the motor, speech motor and the auditory region starting out, but as they age, it seems that they catch up. And that's been shown in a previous diffusion tensor imaging study. And we certainly see that in this data set as well, in the white matter results, where they're showing age-related increases in this superior longitudinal fasciculus on the left side, which connects between the Broca's area, ventral premotor cortex, and the auditory region. So I don't think it's the right side compensating or anything. It's more of a strengthening of the left side that was previously weak that seemed to be contributing to recovery.
Yeah. Okay, we're almost out of time, and thank you so much for leaving us with so much time to have this interesting discussion. I'm going to end with a question about the ethics. So there was a question that came in quite early on about the children seeming to go through a lot, even with your thoughtful design. And is that fair? Is it ethical to put children through so much to understand stammering at the neurological level?
Yeah, thank you so much for that question. And we have put in a lot of thought about this, and we do have to go through very detailed institutional review board reviews to ensure that whatever we do, any discomfort, any thing that the child experiences is balanced out, or even the benefits are much higher than whatever they have to go through. One thing that I can say is that for all of the testing that is being done, we separate these visits, as many times as possible. The speech and language sessions, for example, can be, separated into 3 different visits as they, want, and they're given frequent breaks between the testing. As I said before, our speech language pathologists are just really expert clinicians, and they engage the children. They regularly get letters from the children because they so enjoy working with them. And they come back. So the fact that they want to come back, and we've been able to see them every year for 3 to 4 years, I think it speaks to our ability to not only make this worthwhile, but fun for them. I really appreciate the question. We're constantly aware and try our best to make sure that the benefits outweigh any kind of discomfort or any. There's not a lot of risk involved, but anything costs involved in the study. And the parents really appreciate the fact that we give them a free assessment, a comprehensive assessment of their speech, language, hearing, cognitive test scores, and all of that. So we believe that the benefits really outweigh the costs. And as I said before, the children, I think they truly appreciate that they are the child scientists in this, and they feel that they are contributing to something larger, and they express this to us. So, but anyway, yes, thank you for that question. I don't know if that was sufficient, but
yeah. No, thank you. Thanks. There's so many more comments that I couldn't get round to, and some really lovely complimentary comments about your work. So thanks very much for sharing that with us today.
Which I can't see at all, so but thank you.
Okay, I'm sure we can get them to you. Thanks everyone for listening, and we'll end the session here. Bye.
Thank you so much.